are formally beginning. Welcome, everybody. First of all, I would remind to all our public that this is a webinar. We will have around 30 to 40 minutes of a lecture from Jonathan, and then we will open to a question and answer session. And you're welcome to ask your question by raising your digital hand, as some of you already did, which is perfect. And I will open your microphone, or you can write your question in the chat or in the question and answer box. So now I would like to introduce to you Mr. Charles Canetti. He is a very important lay leader for our French friends. He lives in Paris. He has been a governor of the Hebrew University for 4 years. He's a very, very active member of the board of the French Friends Association. He's married, has 2 children and 5 grandchildren. He has been the CEO of large companies and now owns and manages a consultancy firm. He has already been bitten by mosquitoes, maybe from Rehovot even, and so he feels very at ease with the subjects of this webinar. So please, Charles, the floor is yours. Please introduce our Professor Jonathan.
Thank you, Mara. It's an honor, a pleasure to introduce this webinar. As you said, I am an active member of the board of the French Friends, and I like it. I like it because working to help the university is something that gives me motivation and some excitement. I am in France. France has a special mind, almost pessimistic. And when I speak with you and my friends in the university, it gives me optimism. So it's a pleasure to work for that. It's also a double pleasure for me to introduce Jonathan. First, he works in the Robert Smith Faculty for Agriculture, Food and Environment in Rehovot. Okay, and this is a place I like personally very much, and the French Friends also, because we have funded some project there 2 years ago. And I would just say that this is a place where nutrition and food is very important, and I think a driver for agriculture environment. Okay, it's one of the missions of the university, feed the world. So I advise people to go to Rehovot and visit this faculty. And the second reason is that we had in Paris the pleasure to have Bohbot as for a conference we organized 3 years ago, and it was a real success. We started at 8 p.m. and we finished at 1 a.m. Okay, don't be afraid, gentlemen, and don't be afraid, because now Mara will shorten the time. So it was a pleasure, this conference. So about Jonathan. Jonathan is from French origin. He studied in France, and he worked at a very famous research institute in agriculture named INRA, where he mastered. Then INRA is one of the top 5 in the world. Then he went to the United States to make his PhD and end the studies. And he worked for USDA, that's #1 research institute in the world, the US Department of Agriculture. And then I think it's in 2014, he came to Israel. He came to Israel, where he is now, as I can see. And he worked in the Robert Smith Faculty, and he makes researches on entomology. So I think that he loved insects. Jonathan loved insects since he was very small boy, and probably his passion for mosquitoes came when he was 3 years old, and he was bitten by a mosquito, and he said, "Oh, why me and not my brother?" So all his life, I think he thought, why me? How mosquitoes choose me and not the other one? And probably I think that the participants, they'd have the same experience that either they are the target of mosquitoes or they are lucky enough to have a neighbor and somebody close to them that is the target of the mosquitoes.
A
So the
spouse.
question is, how do mosquitoes, Jonathan, how do mosquitoes choose you
different
and
people?
not me?
I would love to know the answer. I only have ideas about that. And maybe we can discuss this in the Q&A because I'm sure many people have these questions. Let's keep it for the end.
Okay, it's to you, Jonathan.
Huh?
C'est à toi.
Okay. Thank you, Charles. I would like to thank the Hebrew University and the Friends of the Hebrew University for organizing this webinar. I'm going to start sharing the presentation with you. So hold on a second. And here we go. Please quickly tell me if you guys see only one slide filling your screen.
We see the first slide. It's perfect.
Yes, we see the first slide.
Very good. Thank you. So everything that Charles said is true. And I'm going to start with the title slide, obviously. And I would like, with this title slide, to illustrate how mosquitoes and us have more in common than just sharing blood. And I will develop on this idea as we progress through this presentation. So first, let's begin with a few facts about mosquitoes. The good news is it's only 3,500 species. 95% of these species blood feed on some form of animal or another. Of these 3,500 species, 100 species have a strong preference for biting humans. But it goes on, and this business of sucking blood has been going on for 200 million years. They're very good at it. They've developed many different strategies to make sure that they will be successful. They are small, but they can engorge themselves with blood by tripling their own weight. That's quite a feat. Imagine eating how many burgers to triple your own weight. That's what they can do. And they can still fly after that. They have colonized every regions of the world except Antarctica. And when they are not in annoyance, they are transmitting debilitating or even deadly diseases. The kind of visitors that they inject into our bloodstream include viruses such as Zika, yellow fever, dengue fever. They also inject the pathogen that is Plasmodium, which is responsible for malaria, but they also inject worms into our bodies. And this is why, for these multiple reasons, that mosquitoes are considered the most dangerous animals on the planet, killing more people than any other cause of death in human history. We're talking about 52 billion people in the span of 200,000 years since we've been around. So sharks have nothing on mosquitoes. And to prove you that, I'm going to ask you to hold on to your seat and watch this short movie that will get you into the mysterious world of mosquitoes. So it's not really coming to a theater near you, it's coming to your house. And how do they manage to bite us so well? Before they can land on us and bite us, they need to find us. And that's a very dangerous journey for a mosquito to achieve this feat. And they can do it multiple times during their lifetime. And they do it because they have an amazing sense of smell. In fact, you can paint their eyes and they will still smell you out. So they have an amazing sense of smell. But this sense of smell is not only used to detect an animal host. These insects use smell in every aspect of their life cycle. And that includes young, fresh mosquitoes. Before they find a human host, they will need to find a plant host to get their sugar fill, which will fuel their locomotor activity. Once they are ready to take on this young adult life full of sugar, they seek for an animal host. And again, whether it's to find an animal host or a plant host, they use olfaction. Once the female has fed on you, they will rest, digest, and then seek out a suitable oviposition site. And again, they will use olfaction to find that. Now, coming out from the eggs, hatching from the eggs, are larvae. And these larvae now are switching to an aquatic lifestyle, feeding on bacteria, decomposing organic matter. And in these instances, they also rely heavily on olfaction. So how does the nose of mosquitoes look like? So this is a ventral view of Aedes aegypti, the yellow fever mosquitoes that also transmit dengue and Zika. So let's look at this. This is the proboscis. These are the mouth parts getting into your skin and sucking your blood. These long filiform appendages are the antenna. These are the main sites for sensing odors. But they also have these little knobs flanking the proboscis, the mouth parts. These little knobs, which have a little like white dots on the dorsal side, are called maxillary palps. So basically, the mosquitoes have two sets of noses. They have the antenna and they have the maxillary palps. How do they look like in general? And let's dive into these mechanisms of smelling. So these are side views of an adult mosquito and a larva mosquito, mosquito larvae, excuse me. And these are the dorsal view of the adult head and the larval head. And in brown, you can see the antenna. Now, if you zoom in the antenna, you can see that on their surface, they're covered with little hair-like structures. In our world of science, we call them olfactory sensilla. And these sensilla are actually porous. They have pores through which odorants can penetrate inside and make contact with olfactory receptor neurons. And these neurons can recognize these odors. Now, the problem is these odors are usually hydrophobic. They don't mix well with water-based compounds. And these sensilla are filled with a lymph, a water-soluble medium that is incompatible with these odorants. And therefore, for these odorants to reach the receptor on the membrane of these neurons, you need to be transported by odorant binding proteins. And these are globular, water-soluble proteins that have a hydrophobic cavity. So they can interact with these odorants and transport them into the receptor. So now the receptors, that's very interesting. And this is what I've spent many, many years of my research career looking at. So these receptors are sitting on the surface of these neurons, and they're made of two subunits. There is a red subunit and there is a blue subunit. Now the red subunit can interact with odors. And there is one type of red subunit per neuron. That means that each neuron can detect only a subset of odorants. Now, each red subunit is intermingled with a blue subunit. And this is a constant feature of all odorant receptors. This is a co-receptor. And this is, you can think of it as a chaperone. It also acts as an ion channel. What it means is that when the red subunit is activated by an odorant, it tells the blue subunit to open the membrane. And when the membrane opens, there is a change in the electrical property of the cell. And now the neuron, being activated by this change, is able to generate an electrical message that will tell the brain, hey brain, there is a chemical you should pay attention to, and you may want to look at what it is. So what I'm trying to tell you here is that these odorant receptors can be qualified as odorant-gated ion channels. They are ion channels activated by odorants. So back in 2005, the genome of this Aedes aegypti mosquito was released. And what we did, we went through the painful exercise of collecting this little tiny antenna from the head of the larvae and also from the head of the adults. And we, using like a typical molecular biology diagnosis tool, PCR, we were able to find out which odorant receptors are expressed in the larval antenna versus the adult antenna. And you can see that the larval antenna has about 24 receptors. 2/3 of them are larval specific, and the adult has many more. And that's a very interesting difference. They are smelling different in water than they're smelling in a terrestrial environment. We saw a movie of the adult. Let's look at a movie of the larvae. These are the antenna, and these are the brushes of the mouth parts. And basically, these insects are feeding all day and all night long on particles, bacteria, and decomposing organic matter. And these are the little antenna that we dissected. I still remember it. I have PTSD because of that. So our lab is interested in understanding the relationship between olfactory genes, I'm talking about odorant receptor genes, and the olfactory mediated behaviors. So how we've been in the lab trying to explore this relationship is by looking at the molecular mechanisms of olfaction. And for that, we've been doing pharmacological characterization of several of these receptors to understand which receptor interact with which odorant. So in order to discover receptor odorant pairs, but also to look at how drugs may interfere with the function of these receptors. The challenge is not small by any measures, because very few meaningful odorants to the mosquitoes have been identified. We've just begun understanding the mechanisms by which mosquitoes detect odorants. We have a map of these receptors where they are expressed, but we don't know to which odorants they interact with. So the big question is, how does the nose, these odorant receptors, filter a complex chemical environment with a limited number of odorant receptors? So we're talking about 100 odorant receptors having to deal with virtually 27 million odorants estimated to occur in the environment. Not only do we have to understand these relationships, but we have to understand in which context these interactions take place within the context of animal host attraction, the habitat of the host, the context of plant host attraction, and in the context of seeking a suitable oviposition site. So it's not a small task. And for the purpose of this presentation, I will focus on one story that we have developed over the years in the lab. And that's the story of indoles, indolic compounds. And they may look weird to you now, but you are very familiar with these compounds. And indoles form a bridge between humans and mosquitoes because we love these compounds, and so do they. So I need to tell you a little bit of a story about indoles. These indoles are released by flowers and human feces. This is indole. This is the shape of indole, and this is scatol. You can see that they very much look alike. The only difference is that scatol has a small methyl group on position 3. Now, indole, pure indole, will smell like mothballs. And pure scatol is repulsive. It's reminiscent of feces because it is heavily present in feces. Just give me a second. Right. Now, if you mix the two together, you'll start detecting more of a floral note. And if you mix them individually with other floral notes, then they start losing their mothball or feces valence, and they start smelling like flowers. And they've been heavily used in the perfume industry. So these are a few examples where you can find them, but that's not an exhaustive list. There are many more. This is what I could just get like on a quick review. So indoles are ubiquitous. Indole and scatol are ubiquitous. They're produced by plants, they're produced by bacteria, they're present in cheese, meat, many other foodstuff. They're also present in beverages, wine and beer, coffee, all kinds of fruits. But we love it so much that we even use it as an additive in candies, in chocolate, and I just mentioned perfume. Now, indoles are very ancient. They are primal signals. They are as old as life itself. They are synthesized by the 3 biological domains of life, including bacteria, archaea, and eukaryotes, plants. They fulfill many significant biological roles, including communication roles between cells and even between organisms, between species even. So the indole core is used by all these organisms to produce tryptophan, which is an amino acid. It produces also, it is used for plants to synthesize plant growth hormones. And it also used to synthesize plant and animal hormones such as melatonin and serotonin. So this is a very ancient signal. Now, the behavioral meaning of indole and scatole in the life of mosquitoes is unknown. Indoles and scatoles are ubiquitous. They are produced by plants, by human hosts, and oviposition sites. So we don't know in which context these compounds are triggering these insects. Is it in one context, or is it in even multiple contexts, which would be quite fascinating. So first, we started with identifying these receptors. Back in 2010 and 2011, we de-orphanized these receptors. In the genome of Aedes aegypti, you have 3 members of this class of indole-sensitive odorant receptors. We call them indolors for short. There are only 2 members in Anopheles gambiae, the malaria, the vector of malaria. But in Aedes aegypti, there are 3 of them. And they look alike based on their composition, based on their sequence of amino acids. So they look alike. We identified that OR10, which is only expressed in the adult, is a scatole receptor. And we also found out that OR2 was expressed in the larvae and in the adult, and this is an indole receptor. And OR2 loves very much indole, but it doesn't like scatole, and vice versa. OR10 loves interacting with scatole, but it really doesn't care about indole. And look, these compounds are so close. So these insects have retained the ability to distinguish these 2 closely related compounds for 200 million years. So these compounds are very important, and it is very important for these organisms to discriminate between these 2 compounds. So what about OR9? So as I was doing my postdocs, my postdoctoral fellowships, there are certain things I could do and certain things I couldn't do. I wasn't my own boss, but I always wondered what is the odorant that will trigger this receptor. So I had to wait about 10 years to be in a position to ask this question. Which ligand, which odorant is activating OR9? Because OR9 is similar to OR2 and OR10, it should be some type of indolic compound. Which one it is, we didn't know. So I tasked my second PhD student, David Ruel, to look into this question and to explore the function of this OR9 receptor. We use in the lab routinely frog eggs. So we take this frog, we steal the eggs from this organism. These are about 1 millimeter diameter cells. They are big cells that you can see with your eyes, your naked eye. We inject the RNA of this OR9 into the egg. The egg acts as a factory, a protein factory. It produces tons of this receptor on its surface. And with a glorified ampere meter, we can measure the response of the egg, of the receptor, when it is activated by an odorant. And we can measure the current that goes through the membrane with increasing concentration in this particular instance. So this relationship between odorant concentration and response of the receptor can be graphed, and we can measure different things. We can measure the sensitivity of the receptor. We can measure how selective it is. So in terms of sensitivity, you could compare how this receptor responds to odorant 1 versus odorant 2. And if you're interested about selectivity, you can challenge this receptor with a panel of multiple odorants. And so we bought a bunch of natural indolic compounds and we screened this receptor. We said, receptor, what triggers you? So you can see here that we tested over 30 indolic compounds. They all have the same core chemical structure, the indole structure. And what's red is what changes between them. And surprise, surprise, it ended up being scatol. Scatol was the most powerful activator of this receptor. So it was the most potent odorant for this receptor. 5 times more potent than indole. Now that's interesting because the solubility, the water solubility of scatol is about 7 times lower than indole. So it could be that the larvae living in the water has developed a more sensitive scatol receptor because this compound is less soluble in water. But how sensitive is this receptor? So what David did is to compare the response of OR9 when it is exposed to scatol and when it is exposed to indole. And you can see here by looking at these little yellow dots, which represent the sensitivity of this receptor for each of these compounds, that OR9 is about 180 times more sensitive to scatol than it is to indole. So now we are left with 2 scatol receptors. In the larvae, we have OR9, and in the adult, we have OR10. And the larval scatol receptor is 20 times more sensitive than the adult scatol receptor. So here, this is what I'm telling you is that OR9 is so far the most sensitive odorant receptor ever documented. So what are we left with here? We have both the larvae and the adult detect indole using the same receptor. But when it comes to scatol, they use different receptors. So we have resolved the pharmacological puzzle here. Took us about, between their discovery and their functional characterization, 15 years. That's ridiculous. But now we want to understand in which context they operate. In the adult, is it in an animal host context, plant host context, oviposition site context? In the larvae, is it because they are attracted to biofilm of bacteria or by decomposing organic matter? We don't know. So the other thing is, as a side note, very recently, and I just mentioned that these endollergic receptors were mosquito-specific, but with my collaborator, Dr. Jason Pitts at Baylor University, we identified one of these endollergic receptors in the common housefly. So it seems that endollergic receptors may be a conserved features in all insects, which is kind of logical since all organisms can detect indoles. So how do we study the behavioral context of these compounds? For this, we need to be able to track behavior. So we need to be able to take an adult mosquito, a larval mosquito, and look at their response when they're exposed to different odor sources. So we need to be able to measure their trajectory in real time, possibly in 3D, in order to track their trajectories and to quantify the trajectory. It could be that the insect is not interested, it could be the insect is interested, but it also could mean that the insect is somewhat interested. So we need something sensitive enough to be able to measure differences between these behaviors, between these trajectories. So back in 2014, I started at the Hebrew University. I was given a very generous startup fund. I had budgeted everything that I needed, but I didn't budget that thing that you see on your screen. I decided to take a risk. I didn't have pretty much any experience in studying behavior, but I bought the thing. So I designed this custom flight chamber. So here you can see this big box. And this box is attached to a release chamber. So mosquitoes are coming from that side. And on that side, you have an odor source. Now, we can control the air going through this chamber with a clean air delivery system. So we can control relative humidity, and we can control heat also with a heater. We can even control carbon dioxide content. But basically, the system was collecting dust for several years. I didn't mention, but there are also cameras on the top of this picture. But the room that I designed is not big enough. I didn't have enough field of view to take a picture of everything. But 2 years ago, Eviatar Sar Shalom, that you see on the picture here, came to my lab and he said, listen, I'd be interested in developing this system. So basically he picked it up and he developed it, he put it together, and he designed different codes and different scripts to analyze behavior. And I will try to explain in a few minutes, less than a minute, okay, 2 minutes, how it works. So this is the chamber. This is the flight chamber, also called a wind tunnel. It's about 1.5 meter long by 50 centimeters wide, 50 centimeters high. You have infrared lights, so you can operate in total darkness. Mosquitoes do not see infrared light, but we shine this light on them, and the light is reflected from their moving parts, and infrared sensitive cameras can pick up on this signal. And we can track this signal. How well can we track it? Well, we can generate 100 XYZ coordinate every second. That's amazing that we can do this in theory. And then you can project these coordinates onto 3 separate planes. You can look at it from a top view, from a side view, and from an upwind view. And so we can look at the behavior of these insects when they're exposed to different odor sources, an oviposition site, a human hand, a plant, and what have you, a pure compound. You can combine them if you want. Okay, so this is the theory, but how does that work? So the first project was something relatively simple. We didn't use the flight tunnel right away. We used something smaller. We used a small water tank. And that water tank is about 35 centimeters by 25. And we put larvae in there, and we decided to take advantage of a very well-documented behavior in the biology of larvae. They don't like light. When they're exposed to light, they run away. And that's intuitive because they don't want to be seen by potential predators. So what we did here is to look at, we took a top view of their movement. So Y, green over X, and we looked at their behavior when they're in total darkness. And look at them. They're not really traveling along the Y green axis. They're just, hovering, chilling here. What happens when you switch on the light? They run away from the light. The light is coming from down here, from Y0. Okay? So that's quite a dramatic behavior. Now, how do you quantify that? So what we can do is plot these trajectories along the y-axis as a function of time. And you can see that these guys don't travel along the y-axis. And that's what you see here. These are flat lines. These are the 4 larvae. But if you look at the larvae that are exposed to light, they run away. They travel along the green axis, away from the light. When they hit the wall, they can't travel any further. These are the averages. And you can even measure velocity. Velocity integrates direction and speed. And you can see that the velocity is much higher in the case of the mosquitoes exposed to light than in the mosquitoes kept in the dark. So that was quite exciting. And in fact, this happened in the last few months. Now, the next challenge, can we use this system to track insects that are exposed to chemicals? So for that, we used crab food. It's a non-attractant for all kinds of organisms. And here, my PhD student David Ruel and Eviatar joined forces because they're both working on larval behavior. And we used a perfusion system that can inject food or water, either no food on both sides of the container, or food on one side and no food on the other side. So this is the trajectories of 4 larvae exposed to no food on both sides of the container. And basically, they are moving randomly everywhere. Now, what happens when you expose them to food? They wiggle around, but they do find their way to the left side. Now, this is very hard to visualize. So what Aviatar came up with is a heat map. What he did is he converted the density of these different coordinates, like how much time they spend in a given area, and he converted it into a heat map. And this is how it looks. And this is actually reversed. So here corresponds to that. This is when they're exposed to food. So this is that. And this is when they're not exposed to food. So you can see that they're more evenly distributed across the container than they are when they're exposed to food. And how can you quantify that? They don't exhibit difference in speed or tortuosity. Tortuosity is how curved their trajectory is, how straight, how inversely straight they are. So you can see that there is no difference in tortuosity and no difference in speed, but their speed is directional, and that's velocity. So here we did the statistical analysis, and they are different. So they do wiggle around. They do it at the same speed between the two treatments, but they do it in a directional way. Okay, final challenge. Can we track flying mosquitoes? So for that, we went back to our flight tunnel, 1.5 meter by 50 by 50 centimeters. And we looked at a top view and a side view of mosquitoes exposed to clean air without CO2 and mosquitoes exposed to carbon dioxide. So let's start with absence of carbon dioxide. They go in one direction and they move back and they move back. So they hover around. Now let's look at mosquitoes exposed to carbon dioxide, because I think many people are familiar with the fact that carbon dioxide is a mosquito attractant. So let's look at that. So this is the top view and this is the side view. They are exhibiting directional flight. They are very much interested in carbon dioxide. And we can track this. Okay, we can graph that. So these are basically a top view projection of the trajectories of these mosquitoes exposed to clean air versus exposed to carbon dioxide. These represent the mean path. These represent how much they travel along the x-axis, the x-axis being this axis, the green axis, odorants being delivered on one end, mosquitoes being released on the other hand. So they travel along this green axis, very much so. The ones that are not exposed to CO2 do not travel along the x-axis. Now they exhibit significant differences in terms of speed, velocity, and tortuosity. So when you have a tortuosity of 1, you're almost on a straight line. So we can measure all that now. So how, with pharmacological information and behavioral data, can generate knowledge that can improve our lives? Well, if you understand the olfactory code of mosquitoes, you can find out what triggers them, what excites them, what attracts them. So you can design traps that can kill them. But also when you understand the pharmacological properties of these receptors, you can maybe discover drugs that affect the function of these receptors in order to design repellents. So that brings me to the second part of this talk. And I would like to talk about the development of an insect repellent we've been working on in the lab. Many of you are familiar with DEET. DEET is the main component, active ingredient in most insect repellents. Unfortunately, DEET is a topical repellent. It means that it only acts at short range. They still find you, but when they come close to you, a few centimeters away from you, they're confused and they don't seem to finish their behavioral sequence and bite you. And we found out that DEET can actually scramble the function of this receptor. In some cases, DEET can activate a receptor, and in other cases, it can inhibit a receptor. But all these effects seem to be occurring in short range. So here, the dream is to develop a repellent that would act at long range. So for this, Aviatar and I developed a system which is based on design that we made. We 3D printed two rings that can sandwich a protective net. That creates a window into a glove. So the glove is protecting the hand. It creates a window whereby the skin can release odors, and that can attract mosquitoes. And sitting on top of it, you have a chemical holder. And this chemical holder can release a compound or a control or what have you. And we can count how many visits will occur using a camera. And this is how it looks like. So I'm going to show you next a top view inside a cage of mosquitoes exposed to a negative control, a solvent, versus our secret compound. So let's start with the videos. So I think it's pretty clear. The one on the left is attracting mosquitoes. There are no repellents. And this one is our special repellent. And it is not really attracting mosquitoes. I think you would agree with that. If you're interested, please contact Yisum, our technology transfer office. So our candidate repellent is very promising. It exhibits very desirable properties. So if you compare DIT and our magic compound, well, DIT is irritating. It can be irritating. It has an oily feel. It only acts at short range. It has also the property of melting plastic. So if you have an, wristband or anything plastic, it may actually eat up through it. There is a disputed mechanism of action, and large doses are required. For our compound, we know it's volatile, so it has promises in terms of long-range protection. It's non-irritating. It's not oily. The mechanism of action is known. It could be mosquito-specific, and it is generally recognized as safe. So this is really exciting. So in conclusions, I would like to reiterate that mosquitoes exhibit highly sensitive and selective odorant receptors, and that's important for the next and final section of this presentation. They can be targeted by drugs, repellent molecules. So understanding the olfactory code of mosquitoes is a key to manipulate their behavior. And I'm talking about attractants and repellency. Now, for the purpose of the final section of this talk, I want to remind you that these receptors can detect a chemical and they can get activated and they can open up into an ion channel. And this compact arrangement has importance for designing or for taking advantage of this configuration for the development of biosensors, especially when these receptors interact with odorants that are significant to human activities. So despite our ancient conflicts, I would like to argue that mosquitoes can be used for beneficial purposes. And the vision is that I actually presented to the French Friends of the Hebrew University in 2017, just as an idea, was to leverage the pharmacological properties of these mosquito odorant receptors for the design of the next generation of biosensors. So you can imagine a portable device that can detect odors. You can detect like if there is a presence of bacteria on your meat and it sends you a signal, and you should be careful. So indoles play important roles in everyday life. These are a few examples. They are present in plants, in foodstuff. We use it as additives. It's also present in feces, and that's important if you want to take bacteria or feces traces in water supplies. Also, if you want to look at post-harvest of fruit post-harvest, if they're contaminated and you need to react and treat it or throw it away. The sooner you can detect the contamination, the better off you are. In any case, the diagnostic value of indole and scatol is paramount in several economic sectors. So, indole is a common reagent for the manufacture of perfumes, drugs, and pesticides. I also mentioned water quality and food and beverages. So detecting this compound with sensitivity and selectivity at this level can only be achieved for now by biorecognition elements such as mosquito odorant receptors. I've only talked about these indolors for this presentation, but we have others. So how did we bridge fiction to reality? We're not exactly there in reality, but we're getting closer. So in 2014, I joined the Hebrew University. The idea emerges. I pitched it to the French Friends of the Hebrew University in 2017. In 2018, I submitted a few grants to the Ministry of Agriculture and ISUM. I was not successful then. Then I met with Dr. Sefi Vernik. And Dr. Sefi Vernik had the same interest. He works at the Volcan Institute here in Israel, and he is a nanotechnologist and an electrical engineer. And basically, our respective expertise are very complementary. So in 2019, we co-submitted a proposal to the ISF. We were not lucky, but the Ministry of Defense picked up the idea. They funded us for a one-year trial. They have renewed that funding, and we hope that in 2021 we'll be close to results. I'm almost done. So where are we with this idea? Here we want to combine an odorant receptor to a carbon nanotube field effect transistor device. So how successful have we been so far? So we can express these functional odorant receptors. Sefi has been able to prepare small membrane fragments, and that's important because you want them in that configuration to be bound to the carbon nanotube. He has recently also conjugated covalently these receptors to the carbon nanotube, and we are about to test this system against an odor containing scatol. So stay tuned. So conclusions. The mosquito olfactory systems, I hope I've convinced you, is quite sophisticated yet complex. These odorant receptors are highly sensitive and selective. They can be targeted by repellent molecules. And understanding the mosquito olfactory code is not only key to manipulate their behavior, it may be a tool to develop leading-edge sensory technology for the benefit of humankind. And I would like to finish with some acknowledgements and thanks to the Hebrew University of Jerusalem for allowing me to conduct this research. I can tell you how amazing it is to go to work every day with a smile. In particular, I would like to thank Mara Calvo-Viggiovanni from the Division of Advancement and External Relations for organizing this seminar with the support of Marianne Sukperi from External Relations. And I would like to thank the support of YISUM, our technology transfer company, the French Friends of the Hebrew University, Charles and Catherine and Deborah and Lawrence. And this research wouldn't be possible without the financial support of the Israel Science Foundation, the Ministry of Defense, our collaborators, Dr. Jason Pitts and Dr. Sefi Vernik. And this is our lab. Dr. Esti Yakir is a pillar of this lab. Without her, I don't think half of this research would be possible. Dr. Amir Dekel is graduating very soon. He was my first PhD student. David Ruel is doing all the behavior that you saw and the pharmacology also that you saw with OR9. And this is the new batch of students. They are just not being lazy here. They just finished cleaning up the lab every week. Before lab meeting, we organize and clean the lab, and these guys just took out the chairs from the office and they let me take a picture of them. So Eviatar does the behavior. Dor Peretz will be making mosquitoes that are devoid of these endollergic receptors so we can look at their behavior. And Yuri Weiner is now also learning how to do pharmacology. I need to thank mosquitoes for their mysterious world of olfaction and the amazing findings that we've been able to uncover. And that's it. Now I will take questions. I think I went over 2 minutes. Sorry about that.
Thank you, Jonathan. It was fascinating, really. Never thought mosquitoes were such a fascinating world. And so now we're open to the question and answer session. And I see we have already a few digital hands with some questions. So I would open the microphone of Linda Hepner. Linda, you can speak now. Linda, maybe you have to open your microphone too?
Yes, Linda is muted.
Linda, you can unmute yourself. Okay, so Okay.
Okay. I already asked my question about speaking in English because my grandson is watching. That was all right at the beginning. It was a wonderful talk. Thank you very much.
Thank you, Linda. Okay, and now we have Joseph Oscar. Yossi, you can speak now.
Can you hear me?
Internet connection is unstable.
We can
Can
see
you hear me? Can you hear me now?
Yes, we can.
Okay. Mr. Bohbot, I have a question to you. Since we have a very big garden, and about 3 or 4 years ago, the situation of the mosquitoes became unsustainable. And I found one company that is coming to disinfect during the hot season, starting in March until September, once a month. They come, they spray the plants of our garden. And of course, they did not tell me exactly what kind of chemical product they are using because it's a secret, but they only told me that the plants absorb that product and without damaging them. And when the mosquito is landing on the leaves of the plant, it is being simply killed. Can you elaborate on this?
Jonathan, your microphone is closed.
I got cut off from the meeting, so I missed some of it. So I'm not familiar with this technology. I couldn't tell you what chemicals they're using for this.
No, but my question is, it is quite effective because frankly speaking, the number of the mosquitoes disturbing us has dropped dramatically. And of course, it's not 100% complete extinction of the mosquitoes. However, this kind of spray is being absorbed by the plants and the mosquitoes just perish.
So I don't know what they're using. I don't know if they're spraying an insecticide or something. What's happening in Israel with sandflies, and that may be related to this, is that they are using attractive compounds that contain a toxicant. So when mosquitoes land on a surface, on a plant, and this compound is present, it is attractive, it is palatable, and they start feeding on it. And what happens is while they're feeding on it, it's also mixed with a toxicant that will kill them after a few minutes. Again, this is my best guess at what they're doing based on your description, but I have no idea really who is this company and on what technology they are relying on.
Yeah, but my question to you, is this harmful to people?
I wouldn't be able to tell you because I have really no idea what is the chemical composition of their product.
Okay.
Uh-huh.
Okay. Anyway, it's very effective.
Great.
Thank you, Yossi. Thank you. Yes, Charles. Yes.
To complete the answer, we also use such a trick in our garden in the south of France, but I know this is insecticide, and I know this is probably not specific to mosquitoes, so it kills probably. So it's I wouldn't say it's absolutely safe because from what the company said to me, it's not so specific. the insecticide, I don't know how to pronounce. Okay, so that's what Jonathan was saying, is not, in my case, they say it's not so specific to mosquitoes.
Okay.
If I may add something to this, it's called attractive toxic sugar bait. And this system is definitely not mosquito specific.
Okay. We have another question from Anika Hinz, and she's asking which mosquito species were you using for your wind tool study?
So we're using Aedes aegypti.
Okay.
The yellow fever mosquito.
And now Ilan from Holland. He's asking, I'm reading with you, we're wondering whether you have a clue what could be the relevance of your findings for the Ministry of Defense, or is it a sensitive question?
I cannot answer, but I think you might be able to guess why the Ministry of Defense would be interested in mosquito odorant receptors. I can't say.
That's very interesting answer. We'll all think about that.
I have very disappointing answers tonight.
So I see in the question and answer box, a question, how does the activation of the odorant receptor affect the attached carbon nanotube, and how is this signal transduced, maybe I'm not reading right, to your recording device?
Yes. So I'm not a nanotechnologist, so I'm going to try to explain what I received from my collaborator, who is an expert on this subject. So basically, they're using the carbon nanotube as a conductor. And so you can impose a current through the carbon nanotube. When you attach a biorecognition element, such as a mosquito odorant receptor, you are creating a disturbance in the flow. And that disturbance of electron flows can be affected by the activation state of the receptor, because the receptor is a charged molecule, because the receptor is also an ion channel, which conducts ions. So you can affect the local electron charge attached to that carbon nanotube. So every time you have an activation event, you alter the flow of electron going through the carbon nanotube. I hope Sefi would be proud of me trying to explain this.
Okay. Now Linda, she has another question, which I think can be interesting for many, many people. She's saying, my husband and I eat the same food. I get bitten as soon as I go into the garden, and he never. I don't like to use anything that will kill butterflies also. So why this happen?
Yeah, this is the $100 million question. So all of us, and we don't have a good answer. Nobody does. One of the theory is that our skin is covered with a flora of microorganisms. So we have bacteria, we have fungi. For instance, when you sweat, your sweat has absolutely no smell, but very quickly it will be colonized by bacteria on your skin, and they will start releasing indoles and other compounds. So different people have different fauna, and the way this fauna is affected by your diet is not understood. Now, a colleague of mine in Stockholm University, Dr. Nushin Emami, she discovered that Plasmodium falciparum, so the parasite that is transmitted by mosquitoes to us, can affect the metabolism of the red blood cells so that they can release different compounds through the skin that can increase our attraction to mosquitoes. So these parasites can manipulate our smell so that we become more attractive by mosquitoes they will bite us and they will take these parasites so that the mosquito can transmit it to someone else. So that's not, it's somewhat related to your question, but it is very likely that the difference that you observe from one person to another in terms of attraction may be due to microorganisms living on you.
And if I may add a question, sometimes we hear that the type of blood can interfere. Is it true?
There is no conclusive evidence that type of blood affects attraction. That doesn't mean it doesn't happen, but we haven't seen any conclusive evidence of that.
Okay, thank you. So we have time for one more question. If someone wants to raise the digital hands or write it in the chat. Otherwise, let me check if there is something I missed. Okay.
I mean, there is I see Joseph Oscar.
Yeah, he answered his question.
I have another question.
Okay.
I have a colleague of mine who's a chemist, and he says that the mosquitoes, when they suck blood, they look for iron. Is this true?
I can't exclude it. What I know is that what mosquitoes are seeking in your blood are a diet rich in proteins. That's what they're going for. One of the signals that they use to make sure that they have found a stream of blood is adenosine triphosphate. It's ATP. But really what they're going after is red blood cells. They need a diet rich in protein to feed their developing eggs. I'm not familiar with their need for iron. Usually insects have very little need for iron.
Okay. We have a few more questions in the question answer box. What would be a benefit to mosquitoes, and how can we protect ourselves from bites?
Okay, so I would need to understand what you mean by benefit, like in terms of what's the ecological benefit of mosquitoes, or how can we benefit from these insects? So in terms of ecological relevance, on one side, you could argue that mosquitoes fulfill important ecological function. They are food items for many insects and bats and birds and all kinds of organisms. They may be also, to some extent, pollinators. On the other side of the spectrum, you could argue, well, if you get rid of one species of mosquito, it would be replaced by another species of mosquitoes, hopefully not a human biting species. So this is the dialogue here.
Okay, so, and I think the last question is, how do you suggest how we can protect ourselves from bites?
Okay, so there is quite an extensive arsenal of mosquito insect repellents in general. The gold standard of insect repellents is DEET, which I mentioned earlier in my talk. And there are competitors. There are different active ingredients like citronellal or eucalyptol. Usually they are not as good as DEET. Again, DEET is the gold standard. Now, if you're sitting on a terrace and you want to enjoy dinner or lunch outside, I would suggest that probably the best mechanism that you could use to repel mosquitoes at a longer range is a fan.
Like with the wind?
Yes, with the wind. They're not very good flyers. They're very light and fragile. So a fan would do the trick at the knee level. That's especially where they go for.
Okay. So thank you, Jonathan, and thank you, Charles. I would remind all our public that if you have more questions, you're welcome to write to my mail, and I will forward it to Jonathan. And I would like to thank Charles for being with us, for the great introductions, and for being a friend of the Hebrew University from France. And of course, thank you to you, Jonathan. And
thank you.
Good luck with your research, and we'll hope to see what your research brought to the defense minister soon. We're all curious now. And thank you to all of us. We have another webinar on November 25. You will receive the recording of this webinar and the invitation for the next one by mail in the next few days.
Thank you, Mara. Thank you, Charles.
Thank you, Jonathan. Thank you, Mara.
Thank you. Leitraot.
Bye-bye.
Bye-bye.